All right, lecture eight. Lecture eight is about heredity. We are going to be solving quite a bit of genetics problems, and these genetic problems will show up on your next quiz. I do have a review package for you for quiz four, but I'm not going to hand it out today yet, right? Because we're not quite there. So the focus of the first half of the lesson is on the concept of meiosis, which is the process by which our body uses to produce gametes, okay, to produce sperm and to produce eggs. The purpose of meiosis is to enable sexual reproduction. You might remember mitosis is used by some organisms as a way of asexual reproduction. The advantage of asexual reproduction is that you only need one organism. It doesn't require as much energy and it can happen relatively quickly. But the biggest problem with asexual reproduction is the lack of genetic diversity. And so in sexual reproduction, which is what we do, and most animals do it, is to create these sex cells that has shuffled the genes that we have. And in doing so, it creates new variety, okay, in our offspring, okay, genetic diversity in our offspring. So the basis of sexual reproduction is meiosis. Now, you should know by now, we have two sets of chromosomes. Half of it, one set we inherit from mom, and one set we inherit from our father. And because we have two sets of chromosome, we are called diploid organisms. Diploid means two sets. Okay. And to represent diploid, we say 2n, 2n, okay, where n is the number of full sets. N is a full set. How many chromosomes do we have in one full set? Sorry, how many? 26, I heard. 23, that's right, 23. Right? One full set is 23. 23 from mom, 23 from dad, and we have two full sets, which means we have a total of 46 chromosomes. Okay? And when we produce the sex cells, which are called gametes, they are haploid. Okay? It's only one set. Okay? So like I said, a haploid sperm fertilizes a haploid egg, and in the end, you're going to get the diploid back. Okay? In the end, you're going to get the diploid back. All right. So haploid is one set, one set. This is not human, obviously, because there are only three chromosomes. Diploid is two sets, and then you can have, for other organisms, they can have like three sets, which are called triploid, four sets, tetraploid, and then anything beyond, we just kind of call it polyploid. Okay, so strawberries, they have eight sets, octoploid. Yam, sweet potato, they have six sets. Okay, we call them hexaploid. Now, one of the things that bothered me, growing up is how do people make seedless watermelons, right? Because to grow a watermelon, you need a seed. But if the seedless watermelon has no seed, how do you make more seedless watermelon, right? It turns out the answer has to do with what we're talking about. So regular watermelons, the ones with seeds, they are diploid. They have two sets of chromosomes. Okay. There is a special breeding technique that we're not going to go into, but this special breeding technique allows the watermelon to become tetraploid. Okay. So for the regular watermelon, how many sets of chromosomes are there for the gametes? if the regular watermelon is diploid, how many sets of chromosomes in the gametes? One set, right? Because when you produce the gametes, it's always half of what you have, right? So this is going to be N. What about the gametes of these 4N watermelons? 2N, that's right. It's always half, right? So they took the gametes of the 2N watermelon, and then they fertilized the gametes from the 4N watermelon, and 2N plus N is 3Ns. Right? And 3N watermelons are seedless. They are sterile. Why is that? Okay. And this is the key point I'm trying to make here. Organisms with odd number of sets of chromosomes cannot reproduce sexually. Okay. How come? Well, if you have 3N, 3 sets of chromosomes, and you try to make gametes, what's half of 3? 1.5, right? You cannot have 0.5 sets of chromosomes. You're missing a whole bunch, right? So that's why odd number of chromosomes, odd number sets of chromosomes, cannot undergo sexual reproduction. They cannot produce gametes because the number is not divisible by 2. Okay, and that's how you get your seedless watermelons. You can also have yellow watermelons. Have you guys had tried the yellow ones before? Yellow ones are pretty nice. They also have square watermelons. Yeah, they have all kind of weird. They just grow it in a box, okay? So then they turn out to be a cube instead of a sphere. Anyways, I do like my watermelons, so I know a lot about them. All right, moving on. So sexual reproduction, we have a bunch of new terms here. They all start with G. So first of all, we have germ cells. Germ cells, as I told you before, the word germ does not mean, bacteria or dirty or anything like that. Germ cells just means they are cells in the body that will become sperm or egg. Every cell in the body are called somatic cells. SOMATIC, somatic cells, skin cell, liver cell, brain cell, they're all somatic cells. But the cells that will become the sperm and egg, we call them the germ cell. You only find germ cells in the gonads. And gonads. Gonads are reproductive organs. For males, the gonads are testes, or testicles. For female, the gonads are ovaries. So in the testicles, in the ovaries, we have these germ cells, which will undergo meiosis to produce gametes. So once again, gametes are the sperm and egg. All right? Germ cells are found in gonads, and they will undergo meiosis to produce gametes. Are we together? So when the sperm fertilizes the egg, it will form a zygote. Zygote is the first diploid cell that is formed with the union of the sperm and the egg. And that single cell will rapidly divide. 1 becomes 2, 2 becomes 4, so on and so forth. And it will become the embryo, which will implant in the uterus, and then it becomes the baby. And so we were all at one point a zygote. of course, there's one egg, and then there are millions of sperm, and all the sperm is racing to find the egg. And as soon as one is able to insert the genetic material into the egg forms a shield, and no additional sperm can inject the genetic information. Okay, so we won the race. A million of these things racing, and then we made it.
All those sperms have the same genetic.
No. Each sperm will carry different genetic information, as we will see.
Even though it's from the same.
Even though it's from the same father. Okay. Recently, someone showed me this thing. And you can think about it. Okay? I'm just gonna tell you, and then you can think about it maybe for the rest of the class. There were two couples, okay? The husbands are twins, and then the wives are twins. With me so far? And then the meme was like, oh, their kids are also going to be twins. Okay. I'm gonna let you think about that. And then after the lesson, maybe you can tell me if this is true or not, okay?
Wait, you mean twins or do you mean like siblings? Because I thought they would be siblings, right?
That's an interesting point as well. Okay, so we'll come back to it. All right, we're gonna talk about meiosis now. And the first thing you have to know for meiosis is that we start with 2N. And what do we call this cell? Yes, it's diploid. Yes, but what kind of cell is it? Germ cell. Thank you very much. Germ cells are found in the gonads, okay? Testicles and ovaries. And the germ cell is going to undergo 2 rounds of division. Okay? It's going to split once, and then it's going to split again. And in the end, you're going to get up to 4 different cells. Okay? We started with how many chromosomes here, guys? 2, right? And how many chromosomes here? What? For each cell. 1, right? Okay. So you divide it by 2. Half the material. Got it? Okay. Meiosis. Now I have some good news for you. Maybe you like it, maybe you're not. Meiosis is not going to be on quiz number 4. Okay? But it will be on your final test. You still have to learn it though. That's what we're going to be doing now. Right? So this table is from your study guide. Those of you with tablets, you can go find the study guides. If not, you can just do a table quickly. Okay, some of the information you should already know because I have been telling you. So mitosis, how many divisions do we have in mitosis? How many times does the cell divide? That's what I'm asking you. Mitosis? Didn't you guys just have a mitosis quiz last week? Not how many times does the cell divide? No, there are 4 stages, but that's not what I'm asking. Yeah. How many times does you start with 1 cell? 1 time, guys.
1 time. Oh, time. Oh, it's like that's
It's all right. There we go. Okay, the answer is, what's the answer? 1. Okay, once. Thank you. Once. 1 division, right? That's called telophase, by the way. Cytokinesis. What about meiosis? I just told you a slide ago. 2 times, right? Twice, right? So you start with 46. Now this is gonna look weird, but just believe me for now, okay? It's gonna become 23. And then when you split again, it's still 23. Okay, it looks strange, but it's like the way we count them, right? Remember? If they're stuck together, we still count it as 1. Right? So don't get too bogged down by it. Twice. Yeah, this is in study guide number 8. Yeah. Okay. Number of daughter cells. How many daughter cells do we get from mitosis? Take a look at here. 2, right? 2 daughter cells. That's right. Yeah. So because we have to create exact copy of the original, right? Start with 46, you double it, and then you split, it becomes 46. How many daughter cells for meiosis? Huh? Four? Four. Okay, thank you. So it's not as simple as that, it turns out. For men, males, it's 4 sperms. For females, it is 1 egg plus up to 3 polar bodies. I'll tell you what they are later, okay? Just copy it down first. Are the daughter cells haploid or diploid for mitosis? You start with 46, and in the end, they also have 46. So are they haploid or diploid? Okay, one more time. Haploid means 1 set. Diploid means 2 sets. Okay, so 46 is how many sets? Two.
But it's 1 of 2.
No. It's 1 set is 1 to 23. So now you have 1 to 23, and then another one for
Okay.
2 sets. Okay. You guys understand that or no? Yeah, sort of. Yeah.
Of the my that is.
Mm-hmm. Okay. So this is 2 sets, diploid. Okay, maybe I say this one more time. All the cells in the body are diploid, except for sperm and egg. Okay. Germ cells are also diploid, right? But after they divide, then it becomes haploid. The only thing that's haploid, the only thing in your body that's haploid, the only thing in your body that contains 23 chromosomes instead of 46 are the sperm or egg. Okay. Okay, what about meiosis? Diploid or haploid? Haploid. Is this, you're looking for this word? What? All right. Number of chromosomes in daughter cells, I kind of just wrote it down for you. 46, 23. Okay, you need at least 3 colors for this next part for it to make sense. Okay, we're gonna draw prophase of mitosis. You already know it, but we're gonna draw it again so that I can compare the mitosis for you. Compare mitosis and meiosis for you. Okay, so we're gonna have these little pasta looking things. And what are these lines called, anybody? Yeah, that sounds good. And we're gonna draw some chromosomes, okay? This is chromosome 1 from mom, already copied. Chromosome 2 from mom, it's a little bit smaller. And then chromosome 1 from dad. Chromosome 2 from dad. So, what is the name we use to describe these 2 chromosomes? Chromosome 1 from mom and chromosome 1 from dad, what's the relationship? Starts with H. Homologous, that's right. Okay. So in prophase of mitosis, homologous homologous chromosomes are randomly scattered. Okay. They don't find each other. They don't associate with each other. They're just like all over the place. Okay. So even though I'm only drawing 4 chromosomes there, we know there are 46, right? Okay. So it's like having 46 pair of shoes, but then you just throw them all on the floor. They're not matched up. Okay. The right shoe is not necessarily with the left shoe. They're just all over the place, randomly scattered. That's prophase. How many divisions in meiosis? Do you remember?
Two.
2 divisions, right? So the first division, there are prophase 1, metaphase 1, anaphase 1, telophase 1. And then for the second division, same thing, prophase 2, metaphase 2, so on and so forth. Okay, so we are comparing prophase in mitosis with prophase 1 in meiosis. So here is the cell. We will have our centrioles again. And this time, what's really interesting is the homologous chromosomes will pair up. They will find each other. Chromosome 1 from mom, from like your mother, will find chromosome 1 from your father. And they will pair up like this. I'll write it down first, and then I'll explain more. Homologous chromosomes pair up to form something called tetrad. They undergo crossing over to create okay, copy it down and I will explain. That's your Okay, so these are chromosome 1 from mom and dad, and they pair up, and then they form the tetrad. What's the name between this one and this one?
Sister chromatids.
Sister chromatids, very good. Right? Sister chromatids are identical copies of each other, right? Okay. The blue one and the blue one are also sister chromatids. And so what happens is they pair up, and then they're gonna swap genetic information between the red one and the blue one. Okay. This is called crossing over. And so afterwards, okay, crossing over, we're gonna get we're gonna have a little bit of blue on the red, and then there is gonna be a little bit of red on the blue. Okay. So this is called again, crossing over, and it creates genetic variation. You're basically swapping the genes from your mother chromosome with your father chromosome. Okay, think of it like this. You have a deck of cards, red cards and blue cards, okay? And then you are swapping the cards at random. Okay. I'm not gonna swap the A's with the 10. Okay. A's have to swap with A's, and then 10s have to swap with 10s. And I just randomly swap along the way. I don't swap the whole deck. If I swap the whole deck, then I just end up having the same deck, right? You understand what I mean? Because you randomly swap them at different locations, and in the end, you're still gonna have the full deck, okay? But then it's a combination of the red deck and the blue deck. Do you understand? All right, so we're gonna swap the genes, all right? For simplicity, I'm only showing you at the end, but you know it's happening along the entire chromosome. And in the end, we are basically shuffling the genes that we inherit from our parents and then passing half of it to our kids. Do you understand? Okay. So how much are you related to your kids? 50%, exactly, right? 50%. Okay. Now, previously I said 99.9%, right? So when I say 50%, I'm talking about 50% of that 0.1%, right? Okay, if all these percentage gets you confused, you can ask your math teacher about it. Okay? What about your grandparents? Sorry, not your grandparents, like grandparents and grandkids, like what percent? That's right? Because you know, you got half from your each of your parents, but then you're only passing half on, right? Okay. Very interesting stuff. Okay, so this is called crossing over. And this whole thing is called a tetrad, okay? A tetrad. Okay, this is really gonna mess you up. You have to think about it. How many tetrads will we have in prophase number 1? How many tetrads are there going to be? I'm only showing you 2 tetrads here in the picture, but how many are there in an actual human cell at this stage?
That one is still considered one.
Well, yeah, this is called 1 tetrad, 2 chromosomes together. Okay, anyone?
No, I don't like that either.
Huh? How many? 2? For the whole cell? How many? 26? 20, 23, yes, 23, that's right. 23. Okay. This is chromosome 1111. That's 1 tetrad.
Yeah.
And then 2222, right? All the way to 22, 22, 22, 22, right? And then if you're female, females are XX, their 23rd pair will pair up and 45 pair, but not for men. Okay? Men, the X and the Y are not the same. They cannot pair up. You cannot swap genes between the X and the Y.
So we have 23.
That's right, that's right. Like that. You understand? I read something on the weekend. They say the Y chromosome is shrinking, and they predict, you know, eventually it might be gone. The Y chromosome. Yes. Yeah. Very interesting.
Yeah.
Maybe do a Google search and see what the article says. Okay, so next, we have metaphase. Let's do normal metaphase for mitosis. Okay, so in mitosis, we're gonna have the chromosomes lining up in the center. There we go. Okay, some are big, some are small. Okay, so individual. You know this already. I hope you do. You had a quiz on it last time. But over here, instead of the individual chromosomes lining up at the center, we're going to have the tetrad lining up at the center. And don't forget, they already swapped a little bit of their genetic materials. So instead of saying individual chromosomes lined up at the center, tetrads.
Before it crosses over, like for this part, like here, they're both tetrads?
Oh, the
Or,
tetrad means four, right? That like tetra is four, right?
yeah,
So all this means is that there are four pieces.
although in the thing when you're referring to it, it's like when they have each of, like, the blue has some.
Oh,
That's
no,
what-
I mean it's still I see what you're saying, yeah. So it's tetrad before and it's still called tetrad after.
Oh, okay.
It's just that you have swapped the.
Oh, okay.
So over here, the tetrad lining up in the center as opposed to the individual chromosome. Okay, and whether take a look here, please. Whether the red one is on top or the blue one is on top is completely random. Okay, if this one red is on top, the next one again it's random. It doesn't have to be the same, and it doesn't necessarily have to be different, right? So there are additional 23 different configurations. So that it's actually 2 to the exponent 23, which increases the genetic diversity. So that's why, like, you would never, like, if your parents have another kid, I should say, like, that's why your siblings don't look exactly like you, okay? Because there's so many possible genetic combinations, right? From shuffling our genes, okay? They could certainly look alike, but they will never be the same. And the only time you have a genetically identical person as you is if you have a twin, right? Identical twin. Almost there.
Not even twins, so I think.
Identical twins are genetically identical, but they might grow up to look different because of environmental factors. Have you guys heard of the concept of a savior child? Anybody? No, yeah. So sometimes what happens if a family has a child and they are sick with certain diseases, okay, and they require, say, a bone marrow transplant, okay, but there are no suitable donors. Sometimes they would decide to have another child, right, in hopes that they are genetically close enough that they will be able to be the donor, okay. But of course, yeah, so there are a lot of ethical issues here, right? But of course, it's not a guaranteed thing, right? Because every time you shuffle the genes, right, it's not for certain that it's going to work. Okay, almost there. Hang in. Hang in there, people. Okay, so there we go. We're gonna separate them. Okay. Sister chromatids. Separated from each other. And then over here, instead of the sister chromatid separating, we have the homologous chromosomes separating.
What about twins who are different by their output?
Non-identical twins? Yeah, non-identical twins are from two separate sperms and two separate eggs.
Okay.
Yeah, identical twins are one egg, one sperm, and something happens. Nobody really understand how, but it just causes it to causes the zygote to split into two separate zygotes, and then they each develop into a baby.
Okay. Good.
Of course, like you heard of celebrities who have like twins. They sometimes do in vitro fertilization, right? Which allows them to increase the chance of having twins, actually.
What animals same like when a goat pregnant has a
Mhm.
breed? Are they triplets or they're different?
Yeah, that's a good question. Usually not. Yeah, usually they are not clones of each other, right? When a cat has like a whole litter of kittens, they're separate. Yeah, they're not twins. Homologous chromosomes separate. Okay, and that's it. Telophase is the same. And then I'll show you the PowerPoint slides after. So what do you end up having? Genetic relationship of the daughter cells to the parents, identical in mitosis. In fact, you need them to be identical. If they're not identical, that means they contain a mutation. And we saw with mutations, it could be a bad thing, right? Whereas in meiosis, they are genetically different. Genetically different. And we want it to be genetically different. Otherwise, our kids would be, the same as us, which would be no good.
It should be great.
Yeah. Yeah. One of my fears, I'm gonna tell you this story next semester again, but one of the fears my wife had when we had our first kid was like we're gonna bring home the wrong kid. Right. So like she gave me a mission. She's like, when the kid came out, you gotta follow the baby wherever the baby goes. Okay. Of course, in Canada, actually, they never let the baby leave the mother, right? But her sister, who gave birth in Hong Kong, they took the baby all over the place, okay, to do tests and everything. So she was concerned. Anyways, so the joke is like, I'm pretty sure we brought home the right baby because my daughter looks exactly like me, which is unfortunate for her. So there you go. That's the mitosis and meiosis.
True though, that in Canada, because my mom told me that when I was born, because I had like a hole in my heart, so they ended up just taking me out the room.
Well, I mean, I'm sure if they have some emergency, like a hole in the heart, they might have to. Yeah. Yeah, we actually will learn that next semester too. Okay, so that's that. Let's go back to here. I will post the notes if you missed it. Don't worry, everything is on Luminate. Okay, so I'm just gonna go through the slides quickly to show you what we drew. This is meiosis, and it started with germ cells, okay, in the gonads. And then we have the tetrads forming here. And you can see there are exchange of genetic information. Then the tetrad lines up in the center. And we separate the homologous chromosomes. At this point, they're not really homologous anymore because you swap the information, right? But we can still call it that. And then it will go into two separate cell in telophase. Then meiosis 2 is exactly the same as regular mitosis. Okay, you're gonna line them up again in the center, gonna separate them, and then you end up having 4 cells. Okay. So I'm not going to ask you to describe these things or draw them, okay? But you should have a general idea of what the key differences are and you know, the process. Okay, so we did that already. Next, I'm going to show you specifically what happens in male and female. In general, the process of producing gametes is called gametogenesis. Genesis is creation of gametes. Okay. Technically, gametogenesis is same as meiosis. Okay, but it's just like a more specific description, I suppose. So I told you it takes place in the gonads. For men, it is spermatogenesis. Okay. And again, the genesis of sperm. And for female is oogenesis. This is not a typo, it's double O. Okay, oogenesis. Okay. So spermatogenesis is pretty standard. It's just like meiosis, but oogenesis is a little bit complicated. So here we go. Let's take a closer look. On the left here, this is spermatogenesis. We start with a spermatocyte, which is a germ cell. Okay, you don't have to know the name, okay? Which is a germ cell. Germ cell, yeah. So this is in the testicles, okay? And then it will undergo the first meiotic division. And it's already going to be 23 afterwards, okay? Because of the way we count it. And then it's going to go second division, and we will end up having 4 sperms. Okay, 4 sperms. Each is haploid, right? Now, for oogenesis, we start with a oocyte. That's how you say it, okay? Oocyte. And it's also a germ cell. 46 chromosome, and then 23 and 23. Now, what happens over here when we split the cell, when we do division, we have uneven cytoplasmic division during cytokinesis. Right. As you saw earlier in the picture, you know, with the egg and all the sperms around it, the egg is substantially bigger compared to the sperm, right? Because the egg has to house all the nutrients that's required for the zygote to keep on dividing. The sperm doesn't contribute anything other than the genetic information, okay? Did you know the mitochondria has its own DNA, and all your mitochondrial DNA came from your mother? Okay, because the egg is where all the mitochondria are. And so everybody mitochondrial DNA can be traced back through generations of the maternal lineage. Okay? So anyways, because the egg needs to be really big, when we divide it, we want one egg to get a majority of the cytoplasm, and then the other one doesn't really get much. So this is called a polar body. Okay. The polar body might divide, it might not, but ultimately the polar body will be broken down and reabsorbed by the female's body. So in oogenesis, one germ cell will only produce one viable egg. The other 3 polar bodies, they don't do anything. You understand? 4 sperms for men, 1 egg per division. Yeah. So that's the first difference, right? You guys with me? Yeah. Okay, great. This is the second difference. Right? Spermatogenesis is a continuous process after puberty. And when the boy turns into a man, I guess, they will continue to produce sperm up to 200 and 300 million sperms per day. That's a lot. If you do a division, that's like 2,000 to 3,000 per second, but only half of them will become viable. Okay, and this is a continuous process, right? You hear stories that men can have kids up to like 70, sometimes even 8, right? Like celebrities. Yeah, it's kind of weird, but You know, it happens. But for oogenesis, it's a discontinuous process. Okay. The eggs actually start to undergo meiosis when the girl is still a fetus in the mother's womb. Okay, so we have the before birth, the oocyte is going to go all the way to prophase number 1, and it's going to stay that way until puberty, and which means the girl is born with all the eggs they will ever have in their entire life.
What?
Yes, because of this, right? So after puberty, once a month, roughly a month, per cycle, one egg will be chosen to continue to go from prophase all the way to metaphase 2, right? And then if the egg gets fertilized, and only then will meiosis be complete. Now here's the thing, because the eggs are all there at birth, as the woman age, the quality of the DNA in those eggs might degrade over time. There is a direct correlation with the age at which a woman has a baby to the risk of having genetic abnormalities in the baby. So if someone decides to have kids later on in life, they are usually advised to do genetic screening, okay, just in case, right? Of course, if you can afford it, you can always freeze the eggs. Right? I get like the celebrities. I don't know why I keep on talking about celebrities today. Right? Sometimes they freeze the egg, right, at a younger age, and then they decide to have kids later on, right? So some of the key differences between the two processes.
So if, like, you have all the eggs, like, when you're first, do you think, like, I don't know if this is true though, but if, like, birth control can affect that? Because I know a lot of people, when they go on birth control and then they come off, they have trouble having kids.
Yeah, so there are different kinds of birth controls, right, which we'll also learn next semester. But birth control primarily controls the release of the eggs, right? It's not that they're going to destroy the eggs, so it prevents ovulation,
Yeah.
right? So in that regard, it's not really going to affect the quality. But sometimes the birth controls, a lot of them are hormone based. And once your body normal hormonal rhythm is disrupted, sometimes it's hard to get back to the, yeah. Okay, so I'm gonna talk about one problems here that could happen. It can happen in either man or female, but because men, as I show you, the amount of sperms that are being produced is so much greater in quantity compared to the egg, it's less noticeable when an error like this happens. Okay, this is called non-disjunction. In plain English, non-disjunction is the failure of the chromosomes to separate when they are supposed to separate. Okay, so in the 4 steps, prophase, metaphase, anaphase, telophase, which step is the chromosome supposed to separate? Anaphase, that's right. Okay, so if they fail to separate in anaphase 1, then it's called non-disjunction in meiosis 1. If they fail to separate in anaphase 2, then it's non-disjunction in meiosis 2. It's much easier if I show you a picture and explain to you. Okay? Over here, we have the original cell. What do we call the original cell again, please? Thank you. Germ cell. Okay, and then we have the first division. And then this is the second division. And please take a look here. We have tetrad, right? Remember that word? And then they separate, which is good. Everything is as it's supposed to be. And then over here, these two sister chromatids are supposed to separate, right? And again, nothing happens. No mistake. And we have what we call a haploid cell over here. So this one is N, and the sperm is also N, right? Of course, we're not showing the whole set, but N plus N will give you back the 2N, and this is 2N. So over here is fine, is normal, okay? Let's take a look at here. This tetrad, this chromosome failed to separate. We have a non-disjunction here. Failed to separate. And so there is an extra chromosome in this one. This is N plus 1. And then this is N minus 1. It's missing 1, right? So if this is an egg and the sperm fertilizes it, what's the end result? What am I going to write? How many is carried by the sperm? One set, right? N, right? So the egg is N plus 1. If you put them together, how much? 2N plus 1. Okay. What does 2N plus 1 mean? 2N plus 1 means 2 full sets plus an additional one. Okay? Plus an additional one. What do you think this one is?
And
Sorry, what's that? 1N. Okay. Remember, the sperm is N. And then you fertilize the egg. 2N minus 1. 2 full sets, missing 1. Okay. So what happens? Well, 2N plus 1 means there is extra chromosome. Okay. Severity varies depending on the chromosome. Okay. If a person has an extra chromosome 18, then we say trisomy 18. Person has an extra chromosome 21, and it's trisomy 21. You probably heard of trisomy 21, right? What syndrome is that? Down syndrome. That's right. Okay. So over here, though, if it's 2N minus 1, usually not viable. Okay. Having extra chromosomes will allow the baby to develop with some conditions, undesirable conditions. But if it's missing chromosomes, then it is typically not viable. The baby will not grow. Okay. So it will lead to like a miscarriage, for example. Okay, so over here, what about non-disjunction in meiosis 1? Take a look. At the beginning, we have the tetrad, and after the first division, we already have a problem, right? Okay, over here, they didn't separate at all, so these are missing, and there are nothing to separate here. So in the end, you will end up have 50%, that's 2N minus 1, and then 50%, that's 2N plus 1. So if a non-disjunction is to happen, which one is worse? Meiosis 1 or meiosis 2? Yeah, 1, right? Because if it happens 1, all the gametes, all the eggs are going to have problems. Okay? For this example, at least 2 of them, at least 50% of them will be okay. All right? So remember, I told you for female,
wait, none this time doesn't happen with the female. The female.
that's right. So for female, because they have the eggs and then they're suspended in prophase 1, okay, and then as the cycle goes, 1 egg is going to continue. So if this happens later on in life, there is a higher chance of non-disjunctioning occurring, right? But can this happen in spermatogenesis? Yes, but remember, in spermatogenesis, each time there are like thousands of sperms being produced every second, right? So if 1 or 2 of these things is containing an air, right? The chance of that making all the way to fertilize the egg is pretty low. Does that make sense? Okay. Briefly, we're gonna talk about a karyotype. So karyotype was a fill in the blank for the quiz before. Most of you got it right. But a karyotype is an orderly arrangement of chromosomes of an individual. Okay, so here chromosome 1 all the way to chromosome 23. You can see the chromosome size gets smaller and smaller as we go a higher number. X and Y actually use like different criteria which we're not going to go into. But this is a normal karyotype. Okay, this is a men or a female? Men, right? XY. Okay. So you can take your cells, you can take your blood, and then you can prepare a karyotype. Okay, that's what geneticists do. Like they collect cells from the person and then they prepare a karyotype. What kind of information can karyotype provide? Well, it gives you a gross view of your chromosomes. Right? Immediately you can see this person has two of each. Okay, and I can tell this is a male. This is called amniocentesis. Amniocentesis is a process that can be done in pregnant women between 15 to 28 weeks of pregnancy. Okay, how long is a normal pregnancy? 9 months, right? Okay. Someone said 2 years before, and I was like, yeah, you need some sex ed, buddy. Anyway, so the 15 to 20 week is pretty early in the whole pregnancy duration, right? And what they can do here is they can insert a needle through the belly button, okay, and it's guided by ultrasound, so you don't accidentally poke the baby, and they will retrieve a sample of the amniotic fluid. The amniotic fluid is the fluid that surrounds the baby in the mother's womb. Okay. If you're a mother, you might know, you will know, and even if you're not, you might know that before the baby can come out, there's usually what they call like bricks to water. Okay. So the water that comes out is the amniotic fluid. Okay. The amniotic fluid helps keep the baby floating inside the mother's womb, right? So the mother doesn't feel the entire weight of the baby. That fluid contains cells of the baby, right? That's the key point, right? Contains cells of the baby that just kind of washed off from the baby's skin, right? And so when they collect a little bit of the amniotic fluid, they can extract the cells and then they can prepare a karyotype, okay? And in this karyotype, what kind of abnormalities do you notice?
3.
That's right, here's the extra chromosome 21, right? Okay, so this is a diagnostic tool. Right? If there is a family history or if mother is at risk of carrying a child with genetic abnormalities, this is one of the things they could do, and they could do it quite early, right? So parents can, make an informed decisions of how to proceed. There are other tools available should they want to do this even earlier. It is called CVS, which stands for chorionic villus sampling. The chorionic villus is developed from the same tissue that forms the placenta. Okay, and I don't know if you know this, but the placenta has the same kind of cell as the baby. Right? So this one is transvaginal, again guided by ultrasound. They basically take a bit of a tissue of the chorionic villus, and then they prepare the karyotype from it. And this one can be done even earlier. Okay. These procedures are of course not without risk. Because you're after all poking around where the baby is growing, and but then sometimes the risk outweighs the benefit outweighs the risk. Okay.
Wait, so is the fluid like placenta or is that something completely different?
The fluid, this fluid,
Yeah.
the fluid is not the placenta. The
Oh.
fluid is like, so the placenta connects the mother and the baby.
Yeah.
The fluid fills up the entire womb.
Oh, okay. So so why do people say to eat the placenta?
Yeah, because the people who eat the placenta, they believe there are certain growth factor in it that would rejuvenate their skin.
Does it actually? Or
I don't know. I never ate one before. Yeah.
Find out with the cell is you're saying the age of the
Age is a big factor, but there are other risk factors that would increase the chance of having non-disjunction. Okay, like environmental exposure, that kind of stuff.
What kind of exposure?
Yes. So here's-
Environmental exposures like chemicals. Yeah. For example, there are certain mutagens, right, that causes things to mutate, or there are chemicals that would prevent a cell from dividing properly. Those kind of things.
Living next to the chemical facility can
It could be, yeah, could be. Right.
one of the factors. yeah.
That's right. Okay, I think you guys are losing it. Why don't we take 5 and we'll resume afterwards, okay? Okay, we have some questions here. Okay, let's go through this one. You know what? Actually, I don't have a lot of time. Maybe we'll take these up later. I need to move on to this heredity stuff. Okay, so this is important for the quiz number 4. Right? You will have a lot of these genetics problem that you need to solve for quiz number 4. And so let's get to it. Now, much of what we're going to be doing is based on the work of this guy right here. His name was Gregor Mendel. People usually call him the father of genetics because of all the works he has done. Before we go into the actual material, there are some terms I want to go through with you. Some you already know, some new ones. Okay, genes, you should know what a gene is. Okay, by this point in the course, you should know a gene is a section of the chromosome that contains information to make a protein. Okay, and we also should be aware that a gene can have multiple versions. Okay, if you have a gene for eye color, you can have brown eye gene or blue eye gene or green eye gene, whatever. There are multiple versions of a gene. And these multiple versions, we call them alleles. Okay? I have a question for you. If a gene has 5, 6 different alleles in the population, how many different alleles can you have in your genome at most? One more time. This half of you were listening. If there are 5 different alleles in the human population for a particular gene, okay, at most, how many different alleles can you have in your genome?
Wait, if there's 5?
5 different alleles in the population. No, in the population. So, you know, brown eyes, blue eyes, hazel eyes, green eyes, and give me one more eye. Yeah.
In a population of humans?
That's right. So let's say there are 5 alleles available in the population. Huh?
But is
You
there,
only have one.
You can only have one?
No, but like, you can have one of those.
You
Wait,
can't have
are you
one.
saying 5, like 5, like you're saying 5 in the population, okay, but does that mean 5 for myself?
No, that's what I'm asking you. 5 in the
like
population,
5.
but how many for yourself, guys? At most. 2.
2.
Thank you. 2. Okay. Why? Because we are diploid. Okay. We only have slots for 2. You understand? Okay, here is an analogy. I don't know if this works or not. Okay. Remember back in the days we used to have CDs, right? Before Spotify and Amazon and YouTube, whatever. Right? So imagine you have 2 CD racks, okay, and you only have 2 slots for I don't know what band you guys listen to. Backstreet Boys. I don't know if I'm really old. Okay. Anyways, so these bands have many different albums out there, right? Okay, 5, 6, 10 different albums. But how many slots do you have? 2. So you can only buy 2, okay? And then put it in your collection. You understand? Right? So because we're diploid, all right? Anyways, so when we have our genes, let's take a look at the chromosomes right here. We have a picture of chromosome here. And by now, again, you should know, what do we call these chromosomes? What's the relationship? Homologous, okay? Everybody say homologous, right? So this is from mother and this is from father, all right? And remember, on homologous chromosomes, the order of the gene are the same, although the version of the genes might not necessarily be the same, okay? For example, for gene A, I inherited capital A from mom, but lowercase a from dad, all right? If they are not the same, we say heterozygous, okay? Heterozygous or hybrid. Just like chemistry, right? Heterogeneous and homogeneous, and same kind of description. Hetero means different. So this is heterozygous or hybrid. If they are the same, then we say we are homozygous or pure for this gene. Do you understand?
So then how like, you know how people will say like, oh, like it skips a generation?
Yeah, we're gonna get to all
Oh,
that. Yeah. Hold your horses. Yeah. So, yellow some alphabets for me. Give me another gene that's heterozygous. Just yell them out. Heterozygous. A is heterozygous. Something else? G, yeah, different heterozygous. One more. And yeah, K, yeah. You got it, right? If they're different, heterozygous. Another word for heterozygous, hybrid. Okay, hybrid. Okay. Homozygous or pure are the same. Like E, O, yeah. You got it, right? Beautiful. Okay, next. A character. A character is a heritable feature. A heritable feature is something that can be passed on to future generation. Not everything about you can be passed on to the next generation. You get an earring and you have a hole in the ear, your child's not going to be born with a hole in the ear, right at birth. Okay? Something that you acquired throughout life is not heritable. Okay? You dye your hair purple, your baby's not going to be born with purple hair, right? So acquired traits are not heritable. Okay? When we say a character, we're talking about things that can be passed on. And they can be passed on because there is a genetic basis to it. Okay? Only things that has a genetic basis will be able to be passed on to future generation. So a character could be like flower color. Now, a character can have multiple versions, right? And we call those multiple versions a trait. Right? So one more time, characters are caused by genes and traits are caused by alleles of that gene. Okay, this next part is a little bit difficult to explain. I'll try my best. I've never really found a good way to explain this. Okay, so let's say we talk about eye color. Eye color. Okay, please pay attention. If you don't understand this, nothing will make sense afterwards. Okay. Eye color, let's just look at two possibilities here. We're gonna look at brown eyes. Brown eyes, and what other eye color are there? Blue eyes, sure. Brown eyes and blue eyes, okay? So we use alphabets to represent these different traits, okay, these different alleles. So we will say big B is the allele for brown eyes. And then small b is the allele for blue eyes. I'm going to ask you one more time. How many alleles can you have? Two. Okay, so what are the possibilities? These are the alleles. You can only have two. What are the possible combinations? Oh, not the number of combinations. Like, just list them for me. You can have a big B and a... Sure, big B and a small b. What else? A big B and a... Big B, yeah. And then one more? Are those the only combinations? Oh, but small b and big B same as big B and small b, right? Like if I give you a $10 bill and a $5 bill, it's same as if I give you a $5 bill and $10. Do you understand? Okay. So these are the only combinations, right? Okay. All right. So here we go. The person with big B, what is their eye color?
Brown.
Good. So far, so easy. And then small b, blue eyes. Okay. So usually it's like this, right? Like typically people marry within their ethnicity. And so, both of my parents have brown eyes. I inherit the brown eye genes from them. Big B, I have brown eyes. Okay. So sometimes it's possible the parents have different eye color, brown eye and blue eye. And so, the person inherit the brown eye gene from one and the blue eye gene from the other. And they have big B, small b. And the question is, what kind of eye color do they have? That's right, whichever is dominant. So it turns out in genetics, when you have two alleles present at the same time, most of the time, one allele will cover up the other one. Okay, one allele will be dominant over the other one. Okay? The one that's being covered up, we call it recessive. But the one that's covering it, we call it dominant. Okay, with pigmentation, generally, the darker pigment will override, will be dominant over the lighter pigment. Okay, so brown eyes will be dominant over blue eyes. Okay, darker hair will be dominant over lighter hair. You guys understand? Yeah. Okay. So this one is going to be brown eyes.
Then how do people end up with?
Yeah, don't worry, it's gonna come. I promise you, I'll answer all your questions. I know what you're gonna ask. So these here, the genetic makeup, these are called genotype. 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 The genetic makeup. Okay. And over here, these are called phenotype. The manifestation, the appearance, right, of the gene is called phenotype. All right. So here's the thing. Here's the thing. If you see someone with blue eyes, then you know they are little b, little b. Right? Little b, little b. What do we call it? What's the term we use if they are the same? Oh. Were you guys listening 5 minutes ago? Homo what? Zygous. Homozygous. Or if it's too hard, you can say pure, right? Same is pure. Okay, so this is homozygous. If someone has blue eyes, I know for a fact they are little b. I know they are pure. Okay. In fact, the only way for something to have the recessive appearance is if they are pure. Right? If they don't have the pure genotype, then that means a big B is there, and the big B will always cover up the small b, right? Okay. Here's the second point that's important. If you see someone with brown eyes, a lot of us have brown eyes here, then we actually cannot be certain whether the person is big B, brown eyes, homozygous, or big B, small b, right? Even if I look at their parents, I might not know for certain. Their parents can have brown eyes, but it's not for certain that they're going to be big B. Okay. So my point is this. Two people with the same phenotype does not necessarily have the same genotype. Okay, please write that down. Two people with the same phenotype, especially we're talking about dominant phenotype, might not necessarily have the same genotype. Are you with me, everybody? Okay, so we're just gonna do some quick exercise here to set the stage for our next class. Let's say we have a blue eye person. Blue eye, marries a blue eye. A cross means to have kids, okay? What do you think their kids are gonna have? Are they gonna have brown eyes, blue eyes, some brown, some blue? What's going on? All the time or what? All the time. All right, so let's see why. If the person is blue eye, that means they are little b, right? And then they found someone who's also blue eye, little b. Right? When, let's say this is the man, this is the man, this is the woman. When the man produces sperm, what kind of allele are the sperm gonna contain? Big B or a small b? Small b, right? Because that's all he has. He's little b, little b. The only thing he can pass on is little b. What about the eggs of the woman? Little b, right? Okay. So when the sperm meets the egg, 100% guarantee little b for the babies. You understand? So if you have blue eyes, you don't, but if you did, if you have blue eyes, and you really like blue eyes, you want a whole family of blue eyes, who do you have to marry? Blue eyes people. That's right. Okay. Now does that mean brown eyes individuals cannot have blue eye baby?
No.
Hey, well, let's find out. Okay. Let's say we have a big B, and a big B. Okay. So in this situation, the parents are both brown eyes, yes? Okay, what kind of baby is they gonna make? 100% or what? 100% brown eyes, right? 100% brown eye. Okay? But what if the parents are big B, small b, and big B, small b? What kind of eye color do they have?
Blue and brown.
No, brown eyes, right?
Yeah,
Okay.
brown take-
Because the blue allele is being covered up, right? Okay. All right. Now I'm gonna go through this in more details later, but I'm just gonna show you quickly. All right. It's possible that the sperm is gonna have the small b, right? It's possible the egg is gonna have the small b, right? Okay, I'll show you the math later, but in this case, there's a 25% chance it will be blue eyes. Okay. So if the parent has a hidden allele of some other sort, then it's possible, right, for the baby to have a recessive trait that was not present in the parents. You
Why is
understand?
it 25 and not 50?
Yeah, I'll have to show you the math next time.
Oh.
Okay. Sorry, you have a question?
Yeah, okay, four.
We understand?
Yeah.
Okay. So this is the next one is kind of fun. Okay. So these are some traits that are dominant and recessive in humans. Okay. Who can roll their tongue?
Me.
Cool. I cannot. Right. So if you cannot roll your tongue and your spouse cannot roll their tongue, what's gonna happen to all your kids?
No.
Nobody can roll a tongue, right? So if neither of you can roll the tongue and one of your kids roll the tongue, we got a problem, okay? Freckles is dominant, and then this is no freckles is recessive. Again, to preserve a recessive trait, you have to find someone who also has the recessive trait. Okay, then
What
it's
is
guaranteed. Widow's peak is the hairline. Right, so it makes it like a little M.
Okay.
This is straight hair. Okay, and then here, right? If it's dangling, then it's dominant. If it's attached, it's recessive. There's a bunch of things you can look it up. Okay, six fingers are dominant. Right, 5 fingers is recessive. So if you have 5 fingers and you like to keep it that way, you just have to find someone who has 5 fingers.
Wait,
Okay.
no, you have, yours is. Yours is minus 3.
Just one more. Okay. Can you guys do the hands like this? Right, and then just put it together. Put it together. Yeah. Cross your thumbs. Yeah.
Oh,
Just
wait,
do it
wait.
naturally.
Like this or like?
Like this. Okay. Is your right thumb on top of your left thumb, or is your left thumb on top of your right thumb?
Right on top of right.
Left on top of right.
One of these is dominant. I forget which one is which. Okay, and the other one is recessive. Right? It's easy, right? Okay. A lot of things is genetic. If you do this, you can take a look at your hand and you can count the number of chords. 3 chords are dominant. 2 chords are recessive. Do you guys see the chords?
Two?
If you have none, you should go see a doctor. I think that's 2.
1, 2, 3.
Yeah.
Anyways, you can look these up. It's really fun.
Why is it?
All right. You know what? I think we covered enough today. Okay. Thursday? Thursday is very important class. Okay. Don't miss that. You miss that, you're gonna not do well on the quiz. Bye.
And not this.